From the JAMA Network, this is JAMA Psychiatry Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Psychiatry.
Hello and welcome to our listeners from around the world. You're listening to the JAMA Psychiatry Author Interview podcast. I'm your host and web editor, Dr. John Torous. Today we're thrilled to be discussing a new paper just out in JAMA Psychiatry, Polygenic Risk, Psychopathology, and Personalized Functional Brain Network Topography in Adolescence. We're about to be joined by the two corresponding authors of the paper, Kevin Weiss, MBA, and Aaron Alexander-Bloch, MD, PhD, from the Department of Psychiatry at the University of Pennsylvania in Philadelphia, Pennsylvania. We all know that mental illnesses are brain illnesses and have biological basis, but how do these factors all relate to each other? Specifically, what can we learn if we map genetic risk onto personalized brain topography? And how do these brain networks in turn relate to, say, a young person's broad risk for developing mental health symptoms? To learn more, we'll explore these results in a brand new study just out in JAMA Psychiatry. So thank you very much for joining us, Dr. Alexander-Bloch and Mr. Kevin Sun. To get us started, the paper begins by noting that functional network measures may be intermediate phenotypes of psychiatric and genetic risk. Let's break that down into what are these functional networks and how may they be related to genetic risk?
Yeah, sure. So this is Kevin. I'm a MD PhD student working with Aaron in his lab. So the human brain, we know, is organized into these large-scale functional networks that support the brain's ability to carry out its function. So the brain's ability to think, perceive, and initiate and execute action. And differences in these networks have been related to a range of mental illnesses from depression to schizophrenia. But we also know that these networks have a substantial genetic component, that they're heritable. So given those pieces of information, this picture emerges that functional brain network phenotypes could really be a powerful kind of biomarker of genetic risk for poor psychiatric outcomes. So that's kind of what we were getting at by saying that they may be intermediate phenotypes of genetic risk.
And we know that no two brains are exactly the same. So how does this inter-individual variation challenge our understanding between the relationship of genes and brains? Because these networks may be a little bit different between people, right?
Yeah, exactly. We think of the brain being under genetic control. So obviously, for the most part, people's brains are quite similar. They're under natural selection. But like you say, no two brains are the same. No two people are the same. And so that too is in part the consequence of genes. So we want to understand that, how much of inter-individual variation in functional brain networks is due to these kind of genetic factors.
And to understand how the brain is working in these networks, in the paper you use something called precision functional mapping. What is this for listeners less familiar with neuroimaging techniques and work?
We've talked about these kind of functional brain networks, these large-scale networks of brain areas that are carrying out these different brain functions. Like I've alluded to, the organization of those networks, their topography, are pretty much conserved across people. For example, the visual network is pretty much always at the back of the brain, but each person's brain does deviate to some extent in their specific layout. The borders of the regions that make up each network are shifted one way or the other. The networks might be slightly larger or smaller, so precision functional mapping is a way of using a specific neuroimaging modality, functional magnetic resonance imaging, or fMRI, to capture those inter-individual differences in those network layouts.
So it's a way that we really can begin to look at how people's brains may be a little bit different, as you said, understanding that the broad regions are going to be similar across people.
Yeah, exactly. That's right.
Of course, this paper also looks at the genetic aspects of it. And so diving into polygenic risk scores, in the paper you discuss a lot of F1 and F2. What are these for listeners who are less familiar with psychiatric genetics?
This is Aaron. So we know that a lot of risk for psychiatric disorders is due to genetic factors, right? But we also know that there's a lot of overlap between psychiatric disorders in terms of their genetic risk. Like someone's risk for psychosis relates to their risk for bipolar disorder, autism, etc. And so what we were doing with the polygenic risk scores was two things. First, we were using a kind of transdiagnostic perspective. So those two factors relate to different transdiagnostic risk for psychiatric disorders. So not risk for one disorder, but risk for multiple disorders. And then we harness large-scale genome-wide association studies done on hundreds of thousands of people to predict a cumulative measure of risk for each of these genetic factors. And that's what the polygenic risk score for F1 and F2 are.
And what would F1 put someone more at risk for versus, say, F2?
So F1 is capturing common psychiatric symptoms as well as mood disorders. And so you can broadly conceptualize it as capturing mood. And then F2 is capturing more severe psychotic disorders, so things like schizophrenia and schizoaffective disorder.
That's helpful to understand. And one more background question. The paper deals with the P factor, and what exactly is this in the context of the research?
Yeah, so P factor is essentially a concept of overall psychopathology that's used to capture the shared variance across psychiatric symptoms and disorders. So again, we're getting at transdiagnostic psychopathology because we observe comorbidity across diagnoses. And so we're getting at this kind of transdiagnostic picture of disease.
In this case, would higher P factor be something that is not beneficial or a risk factor or more illness?
Exactly. Yes, that's correct.
Given that background, we can put the pieces together where, again, the paper aims to examine how these polygenic risk scores, F1 and F2 for psychiatric illness, relate to the P factor, personalized functional networks, and their heritability in adolescence. So the data that you use from the study comes from the Adolescent Brain Cognitive Development, ABCD study. At a high level, what type of data were you and the team pulling from the ABCD study to answer these questions, linking brain and genes and illness risk?
Yeah, so the ABCD study that you just mentioned, it's a longitudinal study of about 12,000 kids. We specifically are looking at the baseline time point of kids. So these are young kids ages 9 to 10 years old, and we're looking at mental health phenotyping data, genetic data for those polygenic risk scores, and then also functional neuroimaging data. And so those are the three prongs of the types of data we're looking at.
And with this vast amount of data from ABCD, you and the team used some pretty innovative methods in the paper. Perhaps at a high level, you can outline how the methods and analysis worked, and I'll encourage everyone to review the figures in the paper. You and the team put some really helpful content to visually show people what you were doing as well.
So the main novelty, I would say, of the study is this kind of method of precision functional brain mapping, and I guess relating that to genetic factors. And so I would say precision brain mapping is still quite new in the field of functional neuroimaging. And so in defining these personalized brain networks, what we're aiming for is keeping some level of consistency of network layouts across participants while still allowing individual specific patterns to influence network borders, which is what I was alluding to earlier in describing the background on these functional networks. So to that end, we use a machine learning method to derive an individual-specific atlas of networks using each person's fMRI data as an input, while also using a group atlas as a prior. And that way we can balance both individuality of the networks and keep a group-level consistency for interpretability and biological validity.
With those methods, let's jump into some of the fascinating results that you have in this paper. We'll cover the high-level ones. Again, there's so many interesting ones. I encourage everyone to explore the paper. But let's discuss results around heritability for P-factor, again, level of psychopathology in these personalized functional networks, so everyone's individual brains.
So our main interest when we began this project was really in investigating these polygenic risk scores But one of the advantages of this ABCD cohort is the specific recruitment of twin pairs. And twins are a really powerful way to understand the genetic basis of a phenotype because they allow us to disentangle genetic influences of a phenotype from environmental influences. And so because we had these twin pairs, we could run twin pair-based heritability analyses as a kind of separate, independent way to understand the genetic basis of both the P factor and these personalized functional brain networks. And so what we did find was that there is substantial heritability of both the P factor, which was found to be about 50% heritable, and personalized functional network topography, which was on average found to be about 35% heritable.
I guess I'll just jump in and add, in case it's not clear, that heritability, as we're talking about it, like the percentages that Kevin's referring to, basically refer to the proportion of the variability across people that we think is due to genetic factors.
That's a useful explanation of what it is. And we're saying it's about 50% on what you saw.
For P factor, yes. And then for PFN topography across the brain surface, on average, 35%.
You, of course, also have results how the polygenic risk scores, again, that was the F1, F2 we talked about, how those are related to P factor, again, the level of psychopathology and illness people have. So what was the result between genetics and illness?
Those results were interesting because we found a significant association between P factor and F1, which is the more common mood disorder component that Kevin was referring to before. We didn't find an association between P factor and F2, which is the more severe component related to things like psychotic disorders that Kevin was mentioning. And at first, this is a little confusing, but actually it makes sense if you think about when risk for something like schizophrenia manifests, because people who develop schizophrenia tend not to have symptoms of psychosis until late adolescence or early adulthood, and we're measuring these kids at age 9 to 10. So actually, it makes a lot of sense that the relationship is most strongly seen with the more common mood disorder component in this age range, we think.
That's a very clear explanation of why we're not seeing the psychosis symptoms, because those come later in adolescence or early 20s. And the paper has results around how precision functional mapping topography predicts P factor and polygenic risk scores. What are those results?
Like Aaron mentioned, we first looked at how those polygenic risk scores were associated with the clinical factor, P factor, but then we brought in the neuroimaging element of the brain networks. And so what we found is that those brain networks were associated with P factor as well as both F1 and F2. And so what we're seeing are subtle associations, but they are significantly associated. They're meaningful associations across the board. And what is interesting, as Aaron mentioned, is that F2, while it wasn't associated with P factor, it was reflected in the brain. And so what this kind of implies is that this genetic risk for psychotic disorders, maybe prior to it manifesting clinically, it does reflect in a kind of functional brain signature. So it's potentially an interesting kind of diagnostic biomarker, potentially. Early days for that kind of thing, but an interesting result for sure.
As we've been talking about these personalized topographies in brain regions, from looking at these results, which brain regions and networks were most impacted or showing up again and again in your results?
For P factor and F1, or the polygenic risk score for F1, what we found were really similar brain regions. And specifically, it was prefrontal brain regions. And so that's interesting in that those areas have previously been related to functions of emotional regulation, executive function, reward-related processes. And so it's also interesting in that we're seeing these similar regions reflected in P factor and F1. We also saw P factor and F1 were associated. And so we're seeing kind of convergence at the brain level and at the gene phenotype level. And then for F2, we're seeing divergent brain regions. We're seeing, instead of the prefrontal cortex, this temporal parietal junction. And that brain region has been implicated, interestingly, in schizophrenia in prior studies, specifically linked to auditory verbal hallucinations, which is also pretty interesting.
And what's fascinating about your papers, it gives us these brand new and very strong links between genetics and brain and illness, something again we always talk about, we know, but this is such clear and direct evidence. Of the many results in your paper, and we only covered a fraction of them, which do you think clinicians should pay most attention thinking about care, driving care, discussing risk with families or patients?
With this kind of work, we have to be careful not to claim that there are direct clinical implications. I think that our findings in this paper are more pointing towards a future where we can potentially use genetic information and advanced neuroimaging to do a better job of classifying our patients, finding patients that might respond to different medications, even using imaging and genetics as markers in clinical trials in the future. I would be careful in terms of saying that the actual results now have direct implications for clinical care in an individual patient.
Certainly, we could say they have direct implications for driving mechanistic research and understanding how these illnesses are, again, brain illnesses and biological. If focusing on that mechanistic or understanding the brain, which results do you think will move us the most forward in that aspect?
I think that one of the most exciting things about this work is its presentation of this dual brain imaging and genetics perspective that is underrepresented in our understanding of psychiatric illness. There have been a lot of studies showing brain imaging differences related to psychiatric symptoms. And there have been a lot of studies that have shown genetic risk factors related to psychiatric symptoms. But relatively few studies combine them into one approach, and they really have the potential to be mutually informative because understanding genetic risk gives a potentially mechanistic understanding of the differences that we see in imaging, and imaging provides a real life view into the brain of what these genetic risk factors might be affecting. Whereas usually in genetic studies, we really don't understand how genetic risk is manifesting in the brain.
I think that is what makes this work so exciting for everyone. And again, as you said, we may not be able to use it for clinical care just today, but what are the next steps to begin to think about putting some of these results in the future into predictive models?
I think, that in terms of how we're thinking about next steps, there's two avenues we're actively working on. One involves really leveraging the longitudinal nature of the ABCD. So again, we're looking at the first time point of ABCD in terms of thinking about prediction. We're interested in asking the question of, okay, we think of these networks as being fairly stable. They might have some kind of meaningful change across development. And how might we be able to ask interesting questions about predicting network change? And using the fact that we're following these kids across this extended time span gives us this really interesting opportunity to get to the bottom of that kind of question. The other kind of angle we're interested in terms of another kind of way of modeling these networks and understanding the interplay of these different variables, is looking at other types of genetic variants. And so here we're looking at polygenic risk scores. We mentioned these twin heritability results. Like I said, we're seeing these quite high heritability percentages, 50%, 35%. And what we know is that when we look at the underlying genetic variants that compose polygenic risk scores, what are known as single nucleotide polymorphisms, that only composes a small percentage of that twin heritability. So we want to kind of get at other variants. And so we're looking at another type of variant called copy number variants. And so we know that those are also increasing your risk for psychiatric disorders. And so those are really interesting and a ripe opportunity to ask the question, okay, how do those variants, these copy number variants, how are they related to personalized functional brain network layouts? And that's still an open question as well. So those are the two avenues that are actively ongoing in our lab.
Two very exciting avenues.
I was just going to add a slightly different perspective on that. One of my various hats that I wear is I work as a physician in the Penn First Episode Psychosis Clinic. And we're not there yet, but I think with focused effort in the translational domain, because we too often think about research as completely separate from clinical practice and in clinical practice completely separate from research. And if you think now about our patients, I and many people think that there's enough evidence right now to be getting brain MRIs as part of the evaluation of first episode psychosis. And I also think there's enough evidence to get genetic testing on patients with first episode psychosis. It's not really to do the kind of advanced functional mapping that we do in this paper. The reason that brain MRIs are indicated is because sometimes people with psychosis actually turn out to have frank brain pathology, like tumors or vascular malformations or something like that. And the reason that genetic testing might be indicated is because sometimes they have rare genetic disorders that might increase risk for other medical conditions or other things that might really be relevant to the patient outcome right now. But if we can implement those things in the clinic, then we already have this information that allows us to go back to that data and generate and test the potentially more precise predictive models of the future. So I think it's moving what we have into the clinic and translating, I guess, just way more rapidly than we've been able to do in the past.
I'd almost say impactfully. Again, these are pretty interesting results that could have clinical implications very soon. I imagine people would want to know these things about potential risk of how their genes, again, impact their brains in future illness.
Yeah, no, I definitely agree. There's a lot of ethical considerations to take into consideration when you're starting to talk about providing something like a polygenic risk score to a patient right now in terms of clinical practice. But as those continue to be worked out and the models get better, I think we really are going to see the next wave of both genetic and neuroimaging measures implemented in clinical practice in the not so distant future.
That's why it's so exciting that both of you shared these really groundbreaking results. So again, I'm John Torous, and I've been speaking with Kevin Sun and Aaron Alexander-Bloch about polygenic risk, psychopathology, and personalized functional brain network topography in adolescence. To learn more about this research, you can find a link to the article in this episode's description. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode was produced by Shelley Steffens at JAMA Network. Thank you for listening.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.